So, welcome to session one of homeostasis. It's a new unit. So, we're looking at homeostasis. So, we'll be explaining what it is and neurons today. So, two quite big topics. So, first thing we need to do is understand the concept of homeostasis in the body. And really, I think the best way to think of this is just keeping things the same. So, keeping our temperature the same, keeping our pressure the same, blood pressure, that is. It's just trying to keep everything the same. So it's the control of our internal environment, basically. So there's many things that we have to control, but I guess the key things are water levels. So that's our sort of blood pressure, because our blood is mostly water. Um, so how salty it is affects that, how sugary it is affects that. Blood glucose levels, that's really important because we need glucose for cellular respiration, which is the process through which all of our cells get their energy, and keeping our temperature regulated. Also really important because everything that happens in our body, all of the reactions that take place, they involve special proteins called enzymes, and enzymes have a really narrow temperature range at which they work. If we get too hot, they stop working and we die really quickly. So we've really got to work hard to regulate that temperature. So essentially what we've got is maintaining a state of internal balance and stability. And if that dotted line represents, where our temperature should be, for example, 37, or where our blood glucose level should be, the wiggly line is kind of how things tend to be. Sometimes it gets a bit too much, sometimes it gets a bit too low. And then our body has to bring about regulation to keep it normal in the middle. So it's a self-regulation system. We get rise and fall back to normal levels. It keeps not just chemical conditions normal, but physical conditions like temperature. And that's the optimal for us to survive because everything works best at that level. So here's a visual, one way of looking at it. We get some sort of stimulus that produces a change in the variable. Perhaps we get a raise in temperature. This will get detected by a receptor. Got temperature receptors in our skin. We've also got them in our brain. So the input is sent along an afferent pathway to some sort of control center like the brain. And then the output is sent along an efferent pathway to an effector. Now, this could be a muscle which erects the hairs on our arms and legs, for example, and then we get goosebumps. And what that does, getting goosebumps warms us up because it traps some air, and it should help us return to a better temperature if we've got a bit too cold. So it's about keeping things in balance, and the seesaw analogy is kind of quite a good one there. So here's an example of something we need to keep in balance, and that's our fluid levels. So we talked quite a bit recently about extracellular fluid. Our cells need it for everything really because it's got all of the chemicals that are dissolved in it. We need things like oxygen, things like glucose. So it's really important that we get these this fluid into our tissues, into our cells. So for efficient operation of the cell's metabolism, for its physical well-being, its chemical well-being, the extracellular fluid has to be kept controlled. It needs to be the correct temperature. It needs to be the correct pH, the correct osmolality, which is basically how much salt it's got and sugars it's got dissolved into it. It all has to be kept really at a constant state because we need to make sure that water entering and exiting the cells is kept at a sensible level. Don't forget, our cells haven't got a cell wall. So if loads of water goes into them, they will burst. Or if loads of water leaves them, they will shrink and shrivel. So everything has to be kept in a really good balance. That's why, getting dehydrated, for example, makes us really sick and causes us to die. So the extracellular fluid and tissue fluid we've talked about quite a bit recently. Remember, the blood is circulating via all of the tubes in our body, the vasculature, the vessels, the arteries, the veins, the capillaries, the arterioles and venules, which are smaller versions of the arteries and smaller versions of the veins. All of the components from the blood, except for the red blood cells, can leave the capillaries, so they can leave the vasculature and get into the tissues. Loads of that will make its way back into the capillaries and continue around circulation, or we'd have no blood left, but loads of it stays in the capillaries from where it drains into the lymphatic system. And I think it was possibly only last session we were talking about the lymphatic system and how that fluid drains from the tissues into the lymph and gets then poured back into the blood under that bone there, the subclavian vein, with the blood's at low pressure. So we've got this tissue fluid, which contains the white blood cells. It's really important for defense. It's got these platelets that help to clot, but it's also got really important stuff that we need to get around the body, the hormones, the food, like the glucose, the waste materials like the carbon dioxide. We need to keep all of that constant. I'm not going to play this video because the animations don't really help. You need to watch the video with the sound on, but I've kept it in and you guys can watch that if you want to, because it's a nice little video, but it's not a particularly good one with sort of, I don't know, anatomical diagrams or anything. It's more of a cartoon, but it is quite a good one to understand the regulation of balance, essentially, from homeostasis. But we're going to have a look at a good example now, which is really a really good example because it's not happening particularly at a biochemical level. Most of this is happening at a physiological level, so it's stuff that you'd be aware of happening to your body, so regulation of body temperature. One good thing about the regulation of body temperature, to use it as an example, is it involves the nervous system and the endocrine system. So there's two kind of main ways of communicating around the body, sending information around. And that's through our nerves, which is a really quick, fast way, and through our hormones, which is usually quite a lot slower. But sometimes it can be fast. So, for example, adrenaline is a hormone and that acts quite fast. But generally, hormones work like quite slow, like puberty, pregnancy, those sort of things tend to take months rather than short, fast response that you get with adrenaline. But the nervous system is pretty much always fast. So what we do is we use these two systems, the nervous system and the endocrine system, to send information around the body. And together they help with lots of homeostasis processes, homeostatic processes. So together they both help to balance the heat our body generates by metabolic processes. So when we metabolize stuff, so for example, when we use glucose in our body, it generates heat. Respiration gives out heat. So you get hot when you exercise because you're using your muscles a lot, and therefore the cells are respiring a lot more than normal. So you raise your body temperature through respiration. Things warm up through respiration. So we need to regulate that. And we need to keep our body temperature constant. We're not allowed to let it get too hot, or otherwise our enzymes stop working and we die. Similarly, when we get ill, we talked about this, I think, one of the last sessions, very recent session that we did, but one of the responses when we get ill to help contain the infection is to raise your body temperature, because it increases the rate of phagocytosis by your white blood cells. So why that's really good, it's great, it kills off the pathogens. We don't want it to go so high that it kills off us as well, because our enzymes stop working. So it's really important that if we if we raise our temperature because we're exercising or we've got an infection that we're trying to get under control, that we can bring that temperature back down again. It's really important we can regulate it. There's a few ways we can move heat. These are conduction, convection, and radiation. Which is a little bit of a detour into physics for a bit, to remind you about those. So heat can move by or be transferred by conduction, convection, and radiation. They'll only work if the ambient temperature, so that's like the air and the surrounding environment, is less than the skin temperature, though. So you can't lose body heat if it's hotter than your skin. And this is actually becoming a real problem with global heating caused by the climate catastrophe. because people can't regulate their temperature, and they're starting to have issues where they can't. It's just too hot to get your temperature down, and people are just dying because of the heat. If the ambient temperature is greater than the skin's, heat can only be lost by evaporation. So that's a problem. And there's something called wet bulb temperature, and if that gets too high, you cannot cool your body. It just will not happen. So you're dependent on things like air conditioning, and obviously some countries, some areas don't have access to that technology. So that's going to be an ongoing issue over the next decades as the Earth is getting hotter and hotter. We're probably going to see some of the reports that have been out this week actually regarding climate change is we're probably going to see 4 degrees by mid-century, which is a huge concern. We've seen, yeah, it's extremely scary. The more you read about it, it's absolutely petrifying. We were meant to be keeping heating under 1.5. We've gone over 1.5 this year. We're in very dangerous territory already. So heat transfer. One way it can happen is through conduction. And what happens with this is it's conduction. It happens in solids. So it happens really well in metals, and then less well in other solids. So energy is passed from particle to particle. And basically, this is happening by the particles that are touching each other that make up the solid are vibrating. Everything vibrates. Like you don't feel it because it's so on an atomic level, but everything vibrates unless it's at absolute zero, which is minus 273 degrees C. So obviously, we're way hotter than that. So everything vibrates. And these temperature, heat can be passed from one particle to the next by increasing the vibrations. So this is why we make certain substances out of metal, because they're really good at passing heat in this way. Other substances like wood, for example, are really poor conductors of heat, which is why we use them to make certain things like handles on hands. So that's one way of transferring heat. So if it's a non-metal, it's the kinetic energy, the movement, the vibration is the heat. So that is passed from atom to atom through the bonds. So here we've got, I don't know, some structure. It could be any molecule. So it's several atoms joined together to form a molecule, and there's bonds between them. And when you heat up that substance, the kinetic energy, the vibration, the movement is passed from atom to atom along those bonds. And the more it moves, the hotter it is. And that kind of makes sense if you think about it, because we've got a solid object there, and it's going to vibrate and vibrate and vibrate. Eventually, the movement's going to get so big that those atoms are going to start to move apart, and they'll no longer be in that matrix block-like structure. And then we'll have a liquid because the atoms will be able to flow over each other. And then if you keep heating it, it will move so much that those atoms are going to separate out from each other. And that's when you've got gas, because they've completely separated from each other. They're not even flowing apart. They're sort of floating off at that point. So that's conduction, where the heat is transferred through the bonds and the movement of the bonds. And then another way you can transfer heat is through convection currents. So this can happen in liquids and in air. Got an example here that you'd see in a chemistry lab. So you heat a beaker. The heat's obviously transferred to the water at the bottom, and then the bottom bit of the beaker, the water in the bottom beaker, the bit of the beaker is going to be hotter than the water at the top of the beaker. So what's going to happen is it's going to warm up and move upwards. And as the water moves upwards, it cools down and will then fall downwards to the bottom. So if we have a look at this diagram here, the area of the bottom is warmer than the other areas because that's right next to the flame. As it's heated, the particles in the water there will separate from each other because they're vibrating more. So they'll vibrate and they'll move apart. That water will become less dense because the particles are moving apart, and it will move upwards as a result. It will float because it's less dense. So that will move up. The warmer water gets nearer to the top of the beaker. And as it does so, it's going to push the water that was previously at the top of the beaker to the sides. That's going to move down because it's cooler, and it's going to replace some of the water that moved up from here. So you end up with this convection current going around in the water. The cooler water becomes dense and it sinks, filling the space that's left by the rising water. So you get a convection current. And we can also transfer heat by radiation. So that's actually an electromagnetic wave, which is like the light we get from our sun. And you directly transfer heat by radiation waves to another object. And another thing that's important in animals, humans are animals after all, is insulating materials. So materials which are pretty rubbish at conducting heat are good insulators. So an example there is the bird with its feathers. It's fluffed up its feathers to keep warm. It's trapping layers of air in between its feathers there. Same with the polar bear. Really, really thick fur. If you've got cats and dogs, different breeds will have thick fur close to their skin. If you've got something like a husky, they've got very thick fur near to their skin. Really good insulators because the air gets trapped between the fibers. And air, being a gas, is pretty rubbish at conducting heat. And then you've got, the fibers that we make our clothes from as well. So heat transfer around the body. What happens when our body is changing temperature is the brain receives a input of information about our temperature from the blood. There's the region of the brain called the hypothalamus that has thermoreceptor cells, which are basically temperature receptor cells. They're continually monitoring the temperature of the blood as it flows through it. And this is the core temperature. So this is like our blood temperature. Your skin might be colder. There's some cells in the hypothalamus then that respond to changes in temperature, to increases or decreases. And then there's also, as I just suggested, there's receptors in other parts of the body that are involved in temperature regulation. Is some on the skin. The reason there's some on the skin is because that's on the outside of our body and it's the first to change temperature if the temperature changes in our surroundings. And the skin receptors give like an early warning to say, look, it's cold or it's hot. There's going to be a possible change in core temperature. I mean, it doesn't really matter if our peripheral temperature, our skin changes, unless it's like a big change and we get a burn or something. But as long as it's only a small change, it doesn't really matter. But what does matter is if our core temperature changes, that drops or gets too high, because we can die quite quickly then. So how we go about lowering our body temperature then, the muscles in the arterioles relax. So the arterioles are not the big arteries, but the sort of smaller ones as they're getting towards the very fine capillaries. So the arterioles muscles can relax, and that causes vasodilation of the vessels, and that will allow more blood through to the capillaries. So vasodilation is making the vessels bigger. So more blood can get through to the capillaries around the skin, and this can transfer blood near to the surface to the skin, and this will increase the rate of heat transfer about 8-fold. And if we're lowering the body temperature, also our sweat glands start to increase sweat, which evaporates on the surface of the skin, and that removes heat from the body because that evaporation process takes a lot of heat energy. And then we get something called piloerection or piloerection, some people say, but piloerection. And this is where the erector pili muscles, which you can see, well, you can't see actually, they're not on the diagram, but they're found here by the hair. And what they do is they cause the hair muscles to stand up in an upright stance. It says there it's not important in humans, but it does help a little bit. But it's very important in animals like that polar bear, for example. And then the upright projection of the hairs allows them to trap air, insulating the skin. So if you're lowering the body temperature, putting those down would stop that trap. So thermogenesis mechanisms, which generate heat, would be used as well if you were trying to increase your body temperature. Loads of different behavioral things that we do if we're trying to change our temperature, such as changing clothing, for example. If we're trying to raise our body temperature, the arterioles in the skin cause vasoconstriction, so tightening of those holes, and that will stop the blood getting to the surface. So that's why your fingers and toes and nose and stuff get really cold if you go out on a cold day. Your body just stops supplying them with blood, stops trying to keep them warm. If it's a really cold day, your body will just be like, ooh, let's keep that blood in the center around the organs and maintain our body temperature around our core. If we lose a finger or toe through frostbite, we'll survive, but let's not let that core body temperature drop. So shivering, for example, will help us produce heat because shivering is an involuntary contraction of our skeletal muscles. Skeletal muscle contraction, it produces energy. Well, not produces energy, but it transfers energy from the glucose molecules into heat, which raises our body temperature. So in some animals and human infants, there's chemical thermogenesis, which is brought about by secretion of hormones. And obviously you can do stuff like put in layers on if you're cold as well, like your behavioral changes. So if we have a look at this in terms of a sort of flow chart of what happens, and you're at normal body temperature, and for some reason you get hot, could be the weather, could be doing some exercise, could be ill. Your thermoreceptors are going to pick that up, and then your body's going to start to try and cool you down so that your enzymes don't overheat and you die. So one thing it can do is start sweating. That lowers your body temperature because the evaporation of the sweat from the surface of the skin is very energy intensive process. So that's going to transfer some of your heat energy to the environment through the evaporation process. You're going to get vasodilation, which is going to increase the blood vessel diameter near your skin. It's going to take the blood to your skin, which is why you look a bit pink. If you've got white skin, you look a bit pink when you get hot, if you're white European, because there's so much blood moving to the surface, and that heat will radiate off from the blood. And then hopefully those mechanisms work. You see a decrease in body temperature and you return to a normal temperature of about 37 degrees. Let me just check that then. Is there some reason why you sweat in certain places? There's certain places where you have more sweat glands, but I don't actually know 100%, but I think it might be due to something to do with like scent as well, but I don't actually know, to be honest. We're getting up into the big structures. It tends to be not my area of expertise. I'm more of my background is in, my degree is in genetics and my doctorate is in molecular biology. So I tend to know about the molecular processes in a lot of detail, but not necessarily at the larger end of things. But yeah, I think it might be to do with possibly like mate attraction, that sort of thing, but I'm not 100% sure, to be honest. But definitely worth looking up. Okay, so body temperature, here's the other side of it. Normal body temperature, if you get too cold, thermoreceptors again are going to notice this cold temperature. You're going to start shivering, involuntary muscle contraction. Muscle contraction generates heat. You're going to get vasoconstriction, so constriction of those vessels, really stopping the blood getting to your extremities. So your nose, your toes, your fingers, your body will sacrifice them quite happily to keep your core temperature warm. I mean, generally, if you're just out in town shopping and your toes get a bit cold, you're not going to lose, you're not going to lose them through frostbite, but you'd probably have to be climbing up a mountain or trekking to the North Pole to actually lose them through frostbite, You'll also get goosebumps. You can see here, raised hairs on your arms and legs to help trap some air. I mean, it's not massively helpful, but every little bit helps when you're trying to keep warm. And then that should increase your body temperature a little bit, bringing you back to normal, 37 degrees. So what you see, if you look at this diagram side by side, is you're starting to see a wave there of your body regulating your temperature about a normal level, normal conditions. And that's basically what homeostasis is. Your body's constantly working for regulation of temperature, blood glucose, water levels, all of the things that we've talked about. Works very, very hard. And we'll have a look at some of the other ways it regulates over the next couple of sessions as well. So, as we were saying, the nervous system is involved heavily in homeostasis because of a lot of these receptors are nervous system receptors, and the endocrine system is involved. So we're going to have a look at the nervous system now. This shouldn't be too bad because most of this we've looked at because we've had a look at nerve cells, neurons. We've had a look at nerve tissue, which is essentially the neurons and how they sort of interact together. So that most of this we've done, so it shouldn't be too bad, but we are going to have a look at the impulse in a bit more detail. And we're going to need to know a bit more about that for when we do the muscles soon as well. So as we should be vaguely aware by now that the nervous system is made up of the brain, the spinal cords, and the nerves. And what it does is it enables you to control your body and gives you feedback about the world. One important thing about the nervous system is it is fast. It can be extremely fast because you don't even think about it. We talked briefly about the reflex arc before. You touch a hot object, you react before you've even realized you've touched a hot object. And the control is via an electrical impulse, which brings about generally a short-term change. There's some major differences between the hormonal and endocrine system, but there's also similarities as well. With nervous control, it involves stimulation of receptors in the sense organs, which send electrical impulses. So eyes, ears, smell, taste, skin, which is pressure, pain, temperature. And with the hormonal control, we have the endocrine system, which involves the endocrine glands, which produce hormones, which are chemicals, which are released into the blood and carried to target organs where they cause a change. And the impulses in the nervous system are not released into the blood, they're along the nerves, as we know. So nervous system, generally fast response, hormones much slower because it's got to go around the body via the blood plasma. Nervous response is usually very short-lived, and hormonal tends to be longer term. Think puberty, pregnancy, menstrual cycle, but also not always. Adrenaline's pretty quick and short term. Both the systems involve chemicals. The nervous system involves chemicals at the synapse, or synapse as the Americans tend to call it, but synapse. The main difference is the speed. They both involve some sort of stimulus that's detected and the body realizes it needs to make a change. The stimulus is always detected by some sort of receptor, and the effector is the thing that makes the change. So another main difference, chemical versus electrical, and short-term versus long-term. So, as we were saying, brain, spinal cord, nerves, fast, feedback about the world, electrical impulse, short-term changes. The nervous system is linked to the environment via the sense organs that detect the changes in the environment. And each organ has receptors which are sensitive to changes in the environment, each sense organ, that is, so eyes, ears, skin. These are the changes that they're detecting are called stimuli, stimuluses, I suppose, if you weren't using the quite correct word, but you get what I mean, stimuli. So receptors are specialized groups of cell. I've just got a little box in my way, 1 second. Okay, so nervous tissue, we had a look at this image previously. Two types of cells, the neurons, these are the ones that actually do the impulse generation and conduct the impulse from the spinal cord to out to the body. And then there's the glial cells, and the glial cells are like the support cells that help the structures. So the nerve cells are the neurons. This is how we get information through the whole nervous system, central nervous system, peripheral nervous system. Peripheral means outside. So think of peripheral vision. That's when you talked about stuff you can just see on the edge. Yeah. So peripheral nervous system is the sort of outside bit. Central nervous system, literally the central bit, the brain, the spinal cord. And the information goes out via an action potential, which is a wave of depolarization, which moves along the nerve cell. So that all sounds a bit horrible, action potential and depolarization, but it is literally just charged particles move in places, which is exactly what happens when you rub a balloon on your jumper. You're making charged particles move onto the balloon, and then you can stick that balloon on the wall because it's got charged particles on it, an opposite charge to the wall. Does the axon have something covering it for protection? It does. It's covered in cells that make up something called the myelin sheath, and that insulates it. So yeah, you can see it actually there on that diagram. So we've got those long bits on the neuron are called the dendrites. They're the bits that receive the information at their tip and pass the information along the neuron. We're going to have a look at a little video in a minute. The soma, that's the cell body. That's where specifically at this place here, the axon hillock, that's where the sort of decision is made to fire the neuron impulse or not. Something called summation happens. So you have to reach a threshold of, but basically, have you had enough input before you send that signal? You have to reach a threshold before that goes. The axons are very frequently, they're bundled together with other axons, particularly if they're going down a big, like your arm, a big pathway. So there's lots of them bundled together. They're very much like in your house wiring. You don't tend to have just one wire running along for one socket. You've usually got several wires running together, and that's kind of how your body works as well. So the nerves are surrounded by the myelin, which is a lipid. Lipids are fats, and they help to separate so that there's no accidental excitation of neighboring neurons. And then we've got these nodes of Ranvier that allows the signal to jump to speed it up along the nerve. And at the end here, we've got these synaptic terminals. It says presynaptic terminal there because it just means that's the bit before the gap itself is called the synapse. And this is the bit before the gap, basically. So we've got this synaptic terminal, which might be called the synapse or synaptic cleft. It's all the same thing. It's just slightly different names. What happens at the end of this neuron here is you've got a very high concentration of vesicles, or I can't remember what the name. What was the name? There's another name that this course material tends to call them. Vesicles, you get what I mean? They're transport vessels. Vesicles is the name I always use. And basically there's a load of them there because they contain the neurotransmitters that they release into this gap, into this synapse, if the signal is going. So let's just have a quick look at some of these cells because there's lots of specialized cells. So there's astrocytes. They're found in the central nervous system. There's support for the main neurons. What they do is they help maintain something called the blood-brain barrier. Another type of central nervous system cell. These ones produce cerebrospinal fluid. So slightly different role here. But again, it's kind of supporting role. Oligodendrites, these are really important. They myelinate the central nervous system axons and they increase the speed of the electrical impulses. So these are very important support cells. Microglia, again, important support cells. They remove dead and damaged cells. We've got some satellite cells which basically surround and support the neural cells. They're regulating nutrient and waste exchange. And then we've got the Schwann cells, and these are the ones that actually myelinate. These are the ones that form the myelin. These are very, very important support cells. So there's quite a few cells other than neurons, basically. That's what that slide is getting across. You don't need to remember all of those names at all. Probably the most important there are the oligodendrites and the Schwann cells, I would say, because of their role in the myelination, which helps look after the cells. It's very, very hard to remyelinate cells when they're damaged. So, we were talking about injuries a while back and how easy it is for repair and regeneration of tissue. So that myelin is not very easy to replace. So those are important cells there. Okay, let's have a look at the nerve impulse. So as we learned a couple of sessions ago, the nerve membrane has something, well, all cells have a membrane potential. It's basically just how many charged particles they've got inside versus outside. And if there's a difference, so they're not evenly spread out, so there's more positive charged in one area and more negative in another area, as we have on this impulse, then we've got a potential because it's got the potential to change. So what we have often is a difference of between -40 and -90 millivolts. And as I said before, millivolts are quite small. The voltage of the sort of squarish-ish batteries that look a bit about that size, that's about 9 volts. And this is millivolts. The electricity in your sockets is 230 volts. So, -40 to -90 millivolts, that's the difference between one size to another, is pretty small. But when you think how small a nerve is, it's significant. So let's have a look. We're going to zoom in to the axon here of this nerve cell and have a look. What we've got here on the outside is a high concentration of sodium ions. So sodium ions are positively charged. And inside the cell, we've got potassium ions, which are also positively charged, but we've just got loads of sodium outside. So it's more positive compared to the inside. And when the action potential changes, as you can see here, it flips to positive on the inside. The reason that happens is you just get an influx of sodium into the cell. This happens because some channels open and it can move in. So the fact that there's a difference across this neuron membrane of charge, that's what's called an action potential, because there is the potential for action. There is the potential for it to change. So there's a brief depolarization. Essentially, a channel opens, a load of sodium flows inside across the membrane, and then there's a brief positive. You can see it here. It's very quickly returned to negative because they're all pumped back out. There's a brief change to positivity on the inside. And as that happens, that's your neuron being, that your impulse being passed along your neuron. Right. I couldn't actually, the video that I wanted to show you, it wouldn't let me share it. So I've got to stop sharing this, and because I can play it because I've got it on YouTube, but it wouldn't let me embed it for some reason or another. But I'm going to play it to you now to show you that happening. So I'm not going to make it full screen. It's big enough, but I can't make it full screen because I think I might lose it. So those are the dendrites. That's the axon. That's the myelin around it. So this is the myelin here insulating it. Now this is the axon hillock. Now this is like a trigger zone, and this is where the action potential threshold gets built up. So if this nerve gets this neuron gets enough reaches enough of a threshold of stimulation, then it will pass that impulse down this axon, but it has to reach a threshold level because we don't want nerves just firing randomly, or we'd be sort of like having the jitters all the time, we'd be twitching. So we have to have enough of a potential building up, enough of a threshold reached before it signals. So once that threshold has been reached, that impulse goes, and then that goes all the way to the synapse. Look, there's a gap there between the next neuron. The impulse can pass to the next neuron, but it can't jump the gap. It's got to go chemically across the gap, and we'll have a look at that later on. So it can go to the end of the synapse. So here's the membrane of the neuron now. So we'd say this membrane is polarized because there's more positive ions on the outside of the cell compared to the inside. So it's negative by comparison. So this is a pole, like that's what we call it when there's a difference in charge, because it's not the same at each end. So it's polarized because it's different at each end. So there's a difference in charge across the membrane. There's potential for action to occur, or an action potential. So this is a sodium potassium pump, and this is going to be quite important in a minute after the action potential has passed along the neuron, because this is one of the ways we get it back to normal. So what they do is they move 3 sodiums out of the cell and 2 potassiums in. So sodium and potassium are both positively charged. So it's kind of like, well, you're moving some positive out and you're moving some in, but it's always 3 out, 2 in. So essentially, if you do the maths, 3 minus 2, you're constantly moving 1 out, 1 positive ion out. So eventually, you're going to move more out. So that will maintain the charge difference after the impulse. So there's always a little bit of a charge because there's positive on the outside compared to the inside. Essentially, there's more positive outside. So there is a resting potential. It's not kept equal at rest. It's kept ready to go. So it's a resting potential. So if it's going to do anything, this nerve, it detects the stimulus, and then the sodium channel is going to spring into action. What you can see happening there, the sodium channel is opened, loads of sodium is flowing in really quickly. So you're getting loads of positively charged ions moving from the outside of the cell to the inside of the cell. So now the inside is really positive compared to the outside. It's a depolarization. The poles are changed. It was positive on the outside, less positive on the inside. Now it's gone, the positive charges have gone from there to inside the cell. So it's depolarized. The positive end is now inside the cell. So now we've got to get this back to normal because we want to be able to use our nerves again. So we've got to get those positively charged ions, those outside. loads of potassium gets released outside. That's positive, positively charged. It doesn't really do a huge amount getting this potassium outside, but it allows us to use the sodium potassium pump now to pump the sodium back outside. So that potassium that's just been pumped out is used to drive the sodium potassium pump, which pushes the sodium back out. So it's like a wave, that depolarization. If we get enough of a depolarization to hit the threshold level, then that will continue down this axon. Then it gets to the synapse, or it gets to the muscle if it's a neuromuscular junction rather than a neuron-neuron junction. And then we're going to learn in another unit what happens at the muscle then to cause that to contract, because that's a whole other story. So let's pop that slide back up. So if I go back a slide, there you go. You can see that where that positive goes into the cell, what's happening is those sodium ions are rushing across the cell membrane because that sodium channel opens. And when it returns to positively charged on the outside, what's happening is the potassium ions flow back out, and then the sodium potassium pump uses those potassium ions to pump the sodium back out as well. We can see it on here now. So here's our membrane. We've got to move something again. I can't see because this little black box, unfortunately. There we go. So sodium is this green molecule here. Potassium is this triangle, purple triangle. And then we have got a sodium channel. We've got sodium potassium pump, and we've got potassium channel. So what happens is the depolarization is about to take place. The sodium is going to flow in through this open sodium channel. Then, well, this happens first. Actually, I'm going in the wrong direction. This happens first. I've got to follow the arrows. That might help. Then the potassium goes and flows back out. Then this potassium is used as a sort of bargaining chip, if you like, to get the sodium potassium pump working to get the potassium pumped back over there. So we're at a normal potential here at 1. Then we get this huge depolarization as the sodium flows in. Then we get into the recovery phase where we need to pump the potassium out, and then we're back to normal with the sodium potassium pump here. But you'll be able to watch the video actually as well. It's in the course materials if you want to watch that through with the commentary. So resting potential, the inside of the cell is negatively charged compared to the outside because there's loads of sodium, positively charged sodium outside. We get the depolarization, the sodium channels open, sodium ions rush in, and we get that big peak on the graph. Then we have the repolarization, the sodium channels close, the potassium channels open, so the potassium ions go across, making the conditions right for this sodium potassium pump to work and bring about the return to the normal resting potential there. So, what are we doing? This is really important in terms of getting nervous control. We've seen this in the reflex arc. And the really important bit that we're going to look at here is the synapse. And this is the gap between the two nerves. And this is what it looks like, the two neurons. So there's loads of vesicles. These are full of neurotransmitters. And this is how the information gets across the gap between the neurons. These neurotransmitters are released and they diffuse across the gap and they're picked up by receptors. So the electrical impulse can't pass through the gap. The neurotransmitters can, they diffuse across, they combine with the what they're called receptors, and then that will send the signal. We have something called summation happening, and it's how the postsynaptic neuron decides whether to send the signal or not. So the single synaptic impulse is usually synaptic input, sorry, is usually too weak to trigger a response in the postsynaptic neuron. So the neuron after the synapse, it's usually you need to have more than one signal essentially crossing that synapse. So you need to have it either more than one at a time. So we call that spatial summation. So you add, summation means to add together. Spatial summation means they're both working together, or you can have temporal summation, which is over time. So you get multiple signals, if you like, over time. So spatial summation is when you've got neurotransmitters released at the same time, but you've got two in the same space. And then temporal summation is when you've got it building up over time to trigger an action potential in that postsynaptic neuron. So postsynaptic after the synapse. And we've seen this before as well. This is a really important part of the nervous system. So you have a reflex arc, which is your body responding without thinking about it. So stimulus, change in the environment we looked at before was a hot object, pain receptors in your skin. That sends an impulse up your sensory neuron, which then gets to the relay neuron via this synaptic gap. Then there's another synapse here linking to the motor neuron, which will then send the signal to the effector, which is in this case a muscle, but it can be a gland. So the stimulus, the change in the environment, the receptor are the specialized cells that you find in your sense organs, like your skin, your eyes, your ears. Sensory neuron is the neuron that takes the impulse away from the sensor, the receptor, if you like. And then you have the relay neuron, which relays the message to the motor neuron, which is involved in movement at the muscle or the response of the gland, which are both the effectors, the muscle of the gland. These are the things that can respond to the change in the environment. We've had a look at these before. These are the differences in the neurons. So we have sensory neuron there. You can see it's connected to the skin at one end because it's got receptors in it. The relay neuron is short because it's just found inside either the brain or the spinal cord. And then we've got another long neuron there with a motor neuron, because obviously these long neurons have to go down the arms or the legs or somewhere quite long usually. And then this one is connected to a muscle. The stripes tell me it's a muscle because remember that muscle tissue is striated or stripy. And there's lots of important reflex actions that help keep us safe. The startle reflex, so the jump scare, the withdrawal reflex that we just looked at, if you touch something hot or you step on a pin, the iris reflex, which is how your eyes respond if you go into bright light or a dark room, your knee jerk reflex. So that actually stops your knee from collapsing on you every time you take a step. So the reflex arc is really important for walking. Every time you put your weight on your foot, your muscles have to react to support you, and we just flop on the floor without that one. So there you go. We got through it in time. Have you got any questions on that? Okay, if you've got no questions, then I will see you on Thursday, guys.